The crescendo of ovulation, neuroendocrine ballet of menstruation is a masterpiece of physiological choreography. A warm good morning to one and all present here, Honorable Vice Principal Sir, Medical Superintendent Sir, Correlation Seminar Coordinator, Judges Panel, HOD, all the faculty members, resident doctors, and all my dear friends. I am Bharasa Thapa, representing Group B for today's Correlation Seminar. Our topic for today's seminar is Neuroendocrine Regulation of Menstrual Cycle, and our mentor is Khirendra Chaudhary Sir from Department of Physiology. For today's seminar, these are our objectives. With the completion of introduction, we will move on to overview of menstrual cycle. We will also talk about neuroendocrine control mechanisms, which include brain-pituitary-ovarian axis, hypothalamic control, pituitary control, and lastly, we will conclude with dysregulation of the brain-pituitary-ovarian axis and menstrual disorders. Further discussion will be continued by our next member, Christina Raskuti.
Now, let's delve into the basics and the general overview of the menstrual cycle. Menstrual cycle is defined as a cyclical event that occurs in a rhythmic fashion during the reproductive period of the woman's life. On an average, it lasts for the 28 days, but it may be as short as 21 days and as long as 35 days. As the girl enters at the age of the 12 to 15 years, it marks the beginning of her puberty. The onset of the first menstrual period is called menarche, and throughout the reproductive lifespan, the girl undergoes the menstruation and it ceases at the periodical amenorrhea, such as the pregnancy and the lactation, and it continues till the permanent end of the menstrual cycle, which we call as the menopause. As we can see in the slide, towards the left is the ovarian cycle, where there is a regular cyclical growth of the ovarian follicle, which we call as the follicular phase. And towards the post-ovulation, there will be the formation of the corpus luteum, which we call as the luteal phase. Then, there is a right side diagram, which represents the uterine cycle, where the functional endometrium proliferates rapidly, thickens, and shed off in response to the ovarian activity. The orchestration begins in the hypothalamus, where the gonadotropin-releasing hormone sets the stage. This hormone stimulates the anterior pituitary gland to release the two key players, the follicle-stimulating hormone and the luteinizing hormone. Moving to the next slide. Now, we can see in this diagram. We can see there is a follicular phase that is stimulated by the follicle-stimulating hormone. It initiates the follicular development, promotes the growth of the dominant ovarian follicle, and houses the ovum in the ovary. Meanwhile, the luteinizing hormone triggers the ovulation, causing the dramatic release of the master egg from the ovary into the fallopian tube. After the ovulation, there is the transformation of the ruptured graphical follicle from the ovary into a temporary endocrine structure, which we call as the corpus luteum. Now, under the influence of the estrogen secreted by the dominant ovarian follicle during the uterine cycle, there are the marked changes, such as the formation of the new blood vessels in the stroma. There is the endometrial lining proliferation rapidly, and also the uterine gland starts to develop, which we proliferately call as the proliferative phase. This is followed by the secretory phase in the uterus, where there will be the more coiling and the dilatation of the spiral arteries, and there will be more further enlargement on the endometrial lining, which we call as a secretory phase under the influence of the progesterone secreted by the corpus luteum during the post-ovulatory phase. Now, this ovarian release of the estrogen and the progesterone is important for preparing the uterine lining for the potential embryo implantation. If the fertilization does not occur, there is a conversion of this corpus luteum into a temporary corpus albicans, and there is a drop in the level of the progesterone that triggers the menstruation. Now, heading to the next slide. What is the physiology behind the menstrual bleeding? As we can see here, withdrawal in the level of the progesterone, it will upregulate the key inflammatory mediator such as the alpha chemokine CXCL8, beta chemokine CCL2, and the inducible enzyme required for the synthesis of the prostaglandin cyclooxygenase 2. And there is the coincident vasoconstriction of the endometrial spiral arteries that creates the hypoxic atmosphere at the upper endometrial lining, and that will further lead to the release of the vascular endothelial growth factor, which is a vascular permeability factor that further upregulates the matrix metalloproteinase enzyme that creates the tissue destruction, necrosis, and the beginning of the new cycle. Now, the further presentation will be carried by our next member.
Now we will dive deeper into the neuroendocrine dynamics of menstruation. The menstrual cycle is regulated by a complex interaction among ovaries, pituitary, hypothalamus, and even higher centers. The gonadotropin releasing hormone is secreted by a group of neurons in arcuate and periventricular nuclei of hypothalamus. This hormone then travels through the hypothalamic hypophyseal portal system and reaches pituitary, where it causes the release of luteinizing hormone and follicle stimulating hormone. These two gonadotropins through the systemic blood circulation then reach the ovary, where they cause release of progesterone and estrogen. Now these two hormones prepare the uterus for the implantation of a potential blastocyst or menstruation otherwise. They also provide feedback to the higher centers like pituitary and hypothalamus based on their concentrations and the phase of the menstrual cycle. Now we will move forward to hypothalamic control. The GnRH is released in a pulsatile manner. Although this is a cell autonomous property, it is influenced by various neurotransmitters and hormones. For example, there is a group of cells called Kendy cells which secrete dyspeptin, neurokinin B, and dynorphin, which have a stimulatory effect on secretion of GnRH. Similarly, substances like beta endorphin, interleukin 1, and progesterone inhibit the secretion of gonadotropin releasing hormone. Estrogen can have both stimulatory and inhibitory effects. Glutamate and GABA can also influence the secretion of gonadotropin releasing hormone. So what happens in the pituitary now? The released GnRH acts on the gonadotrophs through the GQ receptors, followed by an activation of phospholipase C enzyme. This enzyme then cleaves the lipid in the membranes into inositol triphosphate and diacylglycerol. DAG, being more lipophilic, remains near the membrane where it causes activation of protein kinase C and further activation of many proteins involved in the gene transcription of FSH and LH. The identical alpha subunit but specific beta subunits of FSH and LH then dimerize, are glycosylated and stored within the gonadotrophs. Meanwhile, the other player of this pathway, IP3, causes the release of calcium from the sarcoplasmic reticulum, which is followed by a calcium-mediated calcium influx. Finally, this process culminates in the release of gonadotropins from pituitary gland. As we have discussed so much about the gonadotropin secretion, there still remains a question. As we will see in the slides further, different amount of LH and FSH are released in different phases of menstrual cycle. So, what is the reason behind this differential release? The answer is pulse frequency. When GnRH is released at high pulse frequency, it causes release of luteinizing hormone. But when it is released at low pulse frequency, it causes the release of follicle stimulating hormone. Interestingly, if there is no pulse and GnRH is released at a continuous rate and steady amplitude, it will cause the inhibition of both the gonadotropins. Finally, our secreted gonadotropins reach the ovary where they influence the Graafian follicle. Graafian follicle consists of outer theca cells, inner granulosa cells, and innermost oocyte. The theca cells express the receptors for luteinizing hormone and synthesize androgens by the activation of various enzymes involved in its synthesis. The synthesized androgen then diffuses into the granulosa cells where it is converted into estrogen with the help of follicle stimulating hormone that acts through the GS pathway. Now we will summarize with this beautiful graph here. As you will see on day 26, the FSH level starts to increase. This FSH level then causes the recruitment of various antral follicles which release estrogen and provide negative feedback during the early follicular phase. Eventually, only one dominant follicle remains and it secretes excessive estrogen. As it was asked by an audience member, I need to clarify it. When the estrogen level peaks more than 200 picogram per ml, when it is more than 200 picogram per ml for more than 50 hours, it reverses its pattern into positive feedback and causes the release of LH up to 10 folds and release of FSH up to 2 to 3 folds. This causes ovulation. After ovulation, the graphene follicle remaining in the ovary, the remnant is known as corpus luteum, which secretes high levels of progesterone, estrogen, and inhibin under the influence of luteinizing hormone. If the corpus luteum will decay in 10 to 14 days if there is no conception, causing the shedding of the uterine lining and menstrual blood loss. This summarizes the hormonal regulation in menstruation. Now the presentation will be continued by my group mate Bushra Parveen.
Let's discuss about the dysregulation of this brain pituitary ovarian axis. So we know how this mechanism works. What happens if there is any dysregulation? The brain pituitary ovarian axis helps to control the reproductive function in a female. If there is any dysregulation or if there is any abnormalities of level of hormone, that will lead to pathologies. The pathologies like amenorrhea, specifically hypothalamic amenorrhea, caused by any disturbance in hypothalamus that will impact the secretion of GnRH, eventually impacting the secretion of FSH and LH. Dysfunctional uterine bleeding caused by abnormal hormone levels or any abnormal structure in the uterus, and that will lead to increased bleeding, prolonged bleeding, or excessive bleeding during menstruation of the female. Polycystic ovarian syndrome. That is a complex disorder. It is basically caused by hormonal abnormalities, abnormal GnRH secretion, hyperandrogenism that leads to formation of multiple cysts in the ovaries and infertility of the female. Now the therapeutic targets, the current and future therapeutic targets focus on multiple parts of this axis and helps us to provide the improvement of the condition. Targets like centrally acting agents, GnRH analog, kispeptin, recombinant leptin, they act to modulate GnRH neurons. Gonadotropins acts directly on ovaries, and we have aromatase inhibitors which inhibit the action of aromatase in the uterus and ovary. Now the research on this topic. The current and future research, they basically focus on understanding the intricate interplay between the brain and the reproductive system. The future research may involve exploring how the factors like stress, environmental condition, focus and implement their effect on the axis and the menstruation of female. The advanced technologies like neuroimaging, molecular biology, they help us to understand this complex mechanism much better and provides a room for improvement. Concluding our presentation, understanding the intricacies of brain pituitary ovarian axis provides us a valuable insight on understanding the etiology of the menstrual disorders. And the future research in this topic holds a promise that there will be better management and better enhancement of the health and reproductive function of a female. The references which we used for our presentation are enlisted here. Now talking about the correlation seminar. We students need a strong foundation in our basic sciences to understand and comprehend the complexities of well-being and the diseases in humans. Many students struggle to correlate whatever they learned in basic sciences with their clinical practices. So this correlation seminar is an educational intervention which is designed to enhance our understanding and provides us a platform to correlate our knowledge to our practices. So I would like to thank you on the behalf of team and the department to provide us this platform and this opportunity to increase our knowledge on the subject and become better in this subject. Thank you.